They make it a little easier when you have a study guide. So just for the start of the recording, I'm just going to repeat myself. So you have final exam study guides for neurophysiology on Moodle, pathology, and physiology. You'll find them on your homepage. Okay. So tonight we're going to just be doing an overall review. So this isn't specific to the exam. Your study guide is. Okay, the study guide is specific to the exam. This is just a general review to get you thinking. And I also have another quick review of just the neurophysiological effects of your techniques. Because I know you're starting to learn all those advanced extrinsic techniques. So I just want to do a quick review of that. And that's not really on your Neurophys exam, but I just want to relate them back to what you're doing in practical right now. Okie dokie smoky, so here we go. Review for, so you're going to have to be thinking here because this is interactive. Okay, so first thing, one of the first things we took this year was the anatomy and of the meninges. And so we learned about how the vertebra protects, there's fat that protects, there's these layers of connective tissue that protect your spinal cord. Now they are continuous with the brain, so your entire central nervous system really is protected by the meninges. We saw that there was 3 layers, and they're all made of that dense irregular connective tissue. And they have, you know, on the outer portion there's fat, and then we know there's fluids in between, and that the pia mater, the inner layer, is actually attached right onto the spinal cord and the brain. Okay, so that's just a little reminder of that. So here's your first question. So before you answer in the chat line, just answer for yourself first. Okay, and I'll give you a couple minutes and then I'll start asking you to put it in the chat line. Okay, so first of all, name the 3 layers of the meninges. So for yourself, see if you can name those 3 layers. Now remember, on an exam, you're never going to have to just pull it out of your head like that, right? You'll see the names and have to choose. So name the 3 layers, and then name what is between each layer. So between the dura mater and the arachnoid, what's in there? And then between the arachnoid and the pia mater, what's in there? Okay, and then the last question is, what is the name of the ligament that holds the spinal cord in place? So do you remember the name of that ligament? It goes all the way from the pia mater and out, and it holds it in place from moving side to side. Kind of sounds like it might have something to do with your teeth or getting your teeth fixed. Okay, so what's the outer layer of the meninges? Sorry, Maria, you were just a little tardy to the party. They were answering first on their own. Okay, good. Dura, middle layer. Excuse me, arachnoid. By the way, what does dura mater mean? Tough mother. Don't you just love that? You got a tough mother protecting your central nervous system. Mother's Day's coming up, you know. Okay, and the inner layer I already said was the pia mater. So between the dura mater and the arachnoid, what's in there? Nope. That's in the subarachnoid space. So between the arachnoid and the pia mater, you're correct, Angela, is the CSF. But between the dura mater and the arachnoid layer, so the subdural space. Yeah, in there. So what's in there? No, Leilani, it's fat is in the epidural space, in the outside. So between the dura mater, no, cerebral spinal is in the subarachnoid space. In this subdural is just interstitial fluid, guys. Okay? And then what's the name of the ligament that's holding it all in place? Nope, a good try. No. Filum terminale is what holds it and anchors it down to the coccyx. Okay, so that comes from the pia mater as well, the denticulate ligament. Okay, so let's go back to that picture. See, there's the denticulate ligament. So it comes from the pia mater and comes out and protects the spinal cord from wiggling around side to side. And then the pia mater also comes down and attaches via the filum terminale to the coccyx. Okay, so the three layers again are the tough mother, the arachnoid, and the tender mother. And the tender mother, as you can see, is also very protective because she makes sure it doesn't wiggle around either side to side or up and down. Okay, and then the between the dura and the arachnoid is interstitial fluid, and between the arachnoid, and that's called the subdural space, and in the epidural space outside of the dura is where the fat is. And then in the subarachnoid space between the arachnoid and the pia mater is the cerebrospinal fluid. All right. Of the 4 types of neuroglia within the central nervous system, which one is considered phagocytic? So you can just type this one right into the, good job, Natalie, boom. Okay, so the microglia. Okay, way to remember that, I guess, is it starts with an M, just like macrophages, right? Okay, so we can see the microglial cells right there. There's one there. Okay, so they're there to protect. So they protect the central nervous system from some diseases by engulfing and invading microbes. They migrate to areas of injured nerve tissue where they clear away the debris of dead cells. So they're actually kind of considered the fixed macrophages. Remember, there was wandering, like the monocytes. Okay, they're become wandering macrophages, and then there's some fixed macrophages, like microglia. Of the 4 types of neuroglia within the central nervous system, which one is positioned between neurons and capillaries? Good try, guys, but no, not the ependymal, not the oligodendrocytes. Okay, so here you can see there's a blood capillary. Okay, and there's a neuron, and there's a neuron. Yeah, it's the astrocyte. The process of elimination. Okay, so astrocytes are very busy. Okay, and those of you that were thinking of ependymal, remember ependymal are between your blood and the subarachnoid space and the ventricles and stuff, right? So astrocytes support neurons. They help protect them from harmful substances. They help maintain the proper chemical environment for the generation of nerve impulses. Sorry about that. Assist with growth and migration of neurons during brain development. Blood doesn't get in the CNS. That's what the ependymal cells do, is they filter it, right? And there's blood everywhere to deliver oxygen, okay? But the astrocytes actually help keep tight junctions so that none of the other stuff in blood gets in contact, okay? Yes, astrocytes are in the central nervous system. They're right here, and they're helping keep a tight junction in the walls of these blood vessels. You need lots of blood to the brain tissue for the oxygen. Remember, 2% of your body weight is your brain, but 20% of the oxygen use of your body is used by your brain. Okay, but if you recall, oxygen, carbon dioxide can diffuse right through the cell wall of the blood vessels. Everything else needs increased permeability, which these capillaries do not do in the central nervous system because of the astrocyte. Okay, and then any other blood and all the glucose and stuff we need gets filtered through the ependymal cells into the ventricles, creating the cerebrospinal fluid. Okay, so
Okay,
astrocytes have
thank
huge...
you.
You're welcome.
I really thought that there are no blood capillaries to the brain.
Yeah,
Because,
oh no, there's lots.
Okay, I got
Yeah.
it now.
Okay, good. Yeah, that's important to know because, yeah, you know, you've got a lot of blood coming to the brain. It's called the circle of Willis. Okay, and so there's a lot of blood that comes to the brain, but it never comes in direct contact with brain tissue because of the astrocytes and then, of course, the ependymal cells that filter it and make the cerebrospinal fluid. Okay, so that's called, what the astrocytes do is help maintain that blood-brain barrier. Don't let all the blood get in contact with, but let the oxygen through. All right. Of the 4 types of neuroglia in the central nervous system, which one produces the myelin sheath? Ollie the octopus, right? The oligodendrocyte. Good. So they produce and maintain the myelin sheath around several axons of central nervous system neurons. Okay, so I think it's up to like 15 different axons that they can myelinate. All right, nerve tissue. Where are axon terminals found on the neuron? You're looking at the picture going, it's right there, Sheila. But how would you describe that? At the end, right. And then you can see the axon ends, and then you'll have all these axon terminals at the end, okay, and then they end in a synaptic end bulb. Okay, so the axon and axon collaterals, because there's bigger branches of axons, divide into many fine processes called the axon terminals. All right, back out to the gross anatomy. So here we have the spinal cord and the spinal nerves. Okay, so we know that the spinal cord doesn't go all the way down through all of the vertebrae. We know that it ends in the upper lumbar. We know that there are a bunch of spinal nerves that go and serve the body, and they form plexuses on their way to serve the appendages, the arms and legs. We know that to do that, then the spinal cord has to swell in those parts because there's going to be extra neurons in those sections. Okay, so just a little reminder of all that stuff. When we go inside the spinal cord, we can see the white matter surrounds the gray matter. And in the gray matter is where all the connections are made. And the white matter is obviously representing all the myelination of the axons traveling up and down the spinal cord. We know that we have roots that attach the spinal nerve onto the spinal cord. And the roots also have a function, and they come together, anterior and posterior root, to form a spinal nerve. How many vertebrae were there before the sacrum fused? Oh, boom, you guys know that one. 5, good job. Does the sensory information come in the anterior or posterior gray horn of the spinal cord? Posterior, because a back massage feels good, and we give a massage out the front. So good job. Where is the central canal in the spinal cord? Right in the middle of the H. Good. Right at the center of the gray matter, right? So the central canal. And we know that cerebral spinal fluid also is in there, okay? So there's cerebral spinal fluid in the center, and then, of course, all around in the subarachnoid space. Okay, so 5, posterior gray horn, and the central canal also circulates the CSF. All right, the good old action potential. Which ion is flowing into the cell during depolarization? Okay, so this green line in the picture, which ion is going in? Good job, everybody. So sodium is rushing in. So during an action potential, depolarization to threshold briefly opens 2 types of voltage-gated ion channels. In neurons, these channels are present mainly in the plasma membrane or cell membrane of the axon and axon terminals. The plasma membrane of the axon is actually called the axolemma. First, a threshold depolarization opens voltage-gated sodium channels. As these channels open, about 20,000 sodium ions rush into the cell, resulting in depolarization. The inflow of sodium causes the membrane potential to pass 0 and finally reach +30. Okay, so we know an action potential is really just sodium rushing in. What does the all or nothing principle of action potentials refer to? So if an action potential is going to happen, they say it's all or nothing. What does that mean? Like dominoes, yeah. The strength is always the same, yeah. Once it's happened, right? Okay. So action potentials arise according to the all or none principle. As long as a stimulus is strong enough to cause depolarization to threshold, so -55, the voltage-gated sodium and potassium channels open in sequence and an action potential occurs. A much stronger stimulus cannot cause a larger action potential because the size of an action potential is always the same. A weak stimulus that fails to cause a threshold level depolarization does not elicit an action potential. So, you know, your different stimuli, your graded potentials will be different, right? But once the action potential begins, it is. During an action potential, the ions are moving across the cell membrane by active transport, facilitated diffusion, osmosis, or bulk transport. Okay, so once the sodium is rushing in or the potassium is rushing out, how are they moving across the cell membrane? Facilitated diffusion. Okay, so diffusion is just from high to low. Facilitated means through a gate, right? Simple diffusion goes right through the cell membrane. Okay, and no problem, don't worry about spelling. So we know that sodium, there's lots of it in the extracellular fluid, and so when you open up the gate for it, high to low. So it's really diffusion, but it's facilitated because it has to go through a gate. Okay, so in facilitated diffusion, involving ion channels, ions move down their concentration gradients across the lipid bilayer. Most membrane channels are ion channels, which allow a specific type of ion to move across the membrane through the channel's pore. In typical plasma membranes, the most common ion channels are selective for potassium. So if you remember, it was like 50 to 100 to 1 facilitated channels for potassium over sodium. Okay, or chloride ions. Fewer channels are available for sodium or calcium. Many ion channels are gated. That is, a portion of the channel protein acts as a gate, moving in one direction to open the pore and in another direction to close it. When the gates are open, ions diffuse into or out of cells down their concentration gradient. Gated channels are important for the production of electrical signals by body cells. Okay, so if you recall in your workbook as well as in class, I was bringing you back to chapter 3 when we learned about the different channels, right? So that's from chapter 3, this information. What ion is flowing out of the cell during repolarization? So the red line, which ion is flowing out of the cell? Potassium. Okay, so potassium channels open, potassium ions flow out of the cell, producing the repolarizing phase. Okay, if you recall, I gave you this picture because it wasn't in your textbook. So what is the resting state of the sodium activation gate? What's the resting state? Open or closed? Open. And the inactivation gate? Oh, sorry. No, not open. Sodium activation gate at rest. It's closed, right. So look at the picture here. Resting state. Okay, all voltage gated sodium and potassium channels are closed. Okay, so we can see the activation gate in this picture closed. Yeah. Hey, sorry, I fell for it too. You had me convinced for 2 seconds. Okay, so then the inactivation gate at rest is open. Okay, so at resting state, the activation gate is closed, inactivation gate is open, and the potassium gate is closed. Okay. So each voltage-gated sodium channel has 2 separate gates, an activation gate and an inactivation gate. In the resting state of a voltage-gated sodium channel, the inactivation gate is open, but the activation gate is closed. Oh, okay, well, let's look at it again. So remember that we have to be able to quickly stop the inrushing of sodium. Okay, so when you at resting, it has 2 sections of proteins, really, they're not really gates, that are activated. They're all voltage gated, so they're activated at certain changes in the membrane. So the activation gate is closed until you get a stimulus. So when you get the stimulus of minus 55, the voltage gate, activation gate, is stimulated by that. So at minus 55, the activation gate opens and sodium rushes in. The positively charged sodium goes through that channel and, as we know, brings the inside of the membrane up to plus 30. Plus 30 is the trigger for the inactivation gate to close. Okay, so it closes at plus 30. Okay, plus 30 also is what opens up the potassium gate. Okay, and then as you repolarize and things get to be where they're going to be, it would be in, I think, the second one in the review of the synapse and the review of the action potential. But I also gave you a full sheet with it. One side was the resting membrane potential, okay, and what was open and closed in the membrane to create the resting membrane potential, and on the back was this picture. Okay, and then, yeah, it's definitely in this PowerPoint. Yeah, but just, yeah, I understand your about the names, but yeah, activation gate is the one that opens when you're having an action potential. Okay, so when you have an action potential and it hits threshold, hits minus 55, you're going to have an action potential. That's actually activation gate opens. Okay, and then everything, the gates that open after that are all through this phase here. So minus 55, activation gate opens. Okay, plus 30, inactivation gate closes, and potassium gate opens. Okay, and then as we get back to resting, everybody goes back to their resting. Okay. All right. So in the resting state of a voltage-gated sodium channel, the inactivation gate is open, but the activation gate is closed. As a result, sodium cannot move into the cell through these channels. At threshold, the voltage-gated sodium channels are activated. In the activated state of a voltage-gated sodium channel, both of the gates are now open and sodium goes in. Name the part of the neuron that might be myelinated. The axon. Good job. So in myelinated axons, the voltage-gated sodium and potassium channels are located primarily at the nodes of Grandier, or the neurofibril nodes, the gaps in the myelin sheath. When a nerve impulse conducts along a myelinated axon, current carried by sodium and potassium flows through the interstitial fluid surrounding the myelin sheath and through the cytosol from one node to the next. All right, the good old muscle spindle. Okay, so just a quick reminder, we have extrafusal muscle fibers, which are the skeletal muscle fibers we have been talking about in physiology, right? We also have intrafusal muscle fibers that are wrapped up in some paramysium because they're really, a muscle spindle is really like a fascicle. Okay, so it's wrapped up in some paramysium and it is a receptor in the body, so it's got to have some neurons involved in it. Okay, so it's got two sensory neurons. One is for the reflex, so monitoring a fast stretch of the muscle, and one is for monitoring for tone, so usage of the muscle. Okay, so that would be your secondary, and it's always firing because your muscle is always being used. Okay, because it's part of your muscle and it helps function in setting the tone of the extrafusal muscle fibers, it also, being the only receptor in your body that has it, has a motor neuron into it that can tweak it so that you get a little feedback loop happening to keep your extrafusal muscle fibers firing. The more they fire, the more the spindle fires, the more the spindle fires, the more they fire. Okay, so name the receptors in the muscle spindle. Okay, so the actual nerve receptors. Those are the muscle fibers. Flower spray and annulospiral. Good job. Name the sensory nerve fibers. There are two. So the flower spray, what's its nerve fiber called? Okay. Okay, let's go back then. Okay, so we can see that we have some nerve fibers that wrap around the middle here, right? Okay, so this is an afferent neuron. Okay. Okay. Now the receptor portion would be the annulospiral. Okay, and the its axon would be called a type 1A or primary nerve fiber. Because remember, when we say nerve fibers, we're referring to the axon. Okay, and then we also have, this picture doesn't really show flower spray like it usually is, but more toward the ends is the flower spray, which is the dendrite portion of the neuron. The axon is called a type 2, okay, or secondary fiber. Both of them are alpha, okay, A alpha. Remember there's A, B, and C fibers. A alpha, okay, are for your monitoring proprioception. So they're both going to be the largest myelinated axons, okay, but their names are primary, secondary, group 1, group 2, type 1A, and type 2, depends where you read, okay, and their receptors are called annulospiral and flower spray. The motor to your extrafusal fibers is alpha. So you have the fastest A alpha motor neuron to your extrafusal muscle fibers, and to your inside your spindle, it's a gamma, okay, which would be the third fastest. Okay, so you have the gamma motor neuron goes into your muscle spindle. So you'll hear sometimes, even maybe in exercise concepts, since it was brought up earlier, that you can have gamma gain in your muscles for tone. Gamma gain would be your gamma motor neuron firing more often. And then inadvertently that causes more tone within your muscle when you have gamma gain. Gamma loss would be getting weaker muscles, less tone in them. Okay, but they do, you'll see gamma gain in some textbooks, and that's the gamma motor neuron into the muscle spindle. Okay, so the receptors were the flower spray and annular spiral. You guys had that down pat. The axons are primary, secondary, primary with annular spiral, secondary with flower spray. Okay, and the motor neuron to the muscle spindle we just talked about was the gamma motor neuron, A gamma. And what type of fiber are the intrafusal fibers, muscle or nerve? Muscle. Good job, guys. Okay, so we have annulospiral and flower spray. The nerve fibers themselves are type 1 and type 2, both A alpha fibers. So type 1A is the primary, type 2 is the secondary. We have a gamma motor neuron into the spindle. And yes, intrafusal muscle fibers are actually muscle fibers. So fibers, 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 fibers. All right, the Golgi tendon organ. Okay, a little simpler. Everybody seems not to have as much trouble with it. What is the name of the sensory neuron which transmits information from the collagen fibers at the musculotendinous junction to the spinal cord? One B. Good job, Maria. Okay. It's a type 1B and A alpha fiber from the Golgi tendon organ. So those of you that said GTO, that's not necessarily incorrect. But specifically when we look at it, this neuron coming from it, okay, GTO is referring to the collagen fibers, the connective tissue capsule, and the nerve fiber. Okay, the question was saying what is the sensory neuron? Okay, so that would be the type 1B. A for spindles, B for GTO. Yes. Okay. So type 1B, and it's an A alpha because it's one of your proprioceptors, so it functions in one of your reflexes. So again, like any sensory fiber coming from the GTO or the muscle spindle, they're all going to be the fastest nerve fiber in the body. Okay, and then they just name them kind of like you have muscles throughout your body, but they all have different names, right? Okay, so in this case, they get very specific about these neurons. Not all neurons get specific names like that, like individual neurons, but in the case of the spindle and the GTO, they do. All right, some more simple, way back to the very beginning, just the divisions of the nervous system. Okay, so this is a different kind of chart. That's why I included it, because you have good ones in your textbook as well. But the nervous system, we know, gets divided into a central nervous system, which is your brain and spinal cord, and the peripheral nervous system, your cranial nerves, and your spinal nerves. Now, of course, we have afferent or sensory neurons that send all the information in, okay? And for both the somatic and the autonomic. But efferent, we can look at the two divisions, somatic and autonomic. Somatic is very easy. It has its information coming in from proprioception and from your, mechanoreceptors and pain receptors and stuff, and the output is to only your skeletal muscles. Whereas the autonomic nervous system, the motor division, gets divided into sympathetic and parasympathetic, which we learned about what just briefly was the sympathetic is your freeze, fight, or flight, and parasympathetic is rest and digest, and that serves your cardiac muscles, your smooth muscles, and your glands. Okay, I also just included again, there's not a ton on the exam about the different fibers, okay, but just another reminder, we have A, B, and C axons in the body. They are called nerve fibers, okay. The smallest of those nerve fibers is unmyelinated, and they carry things like slow pain and warm temperatures. Your B fibers are about mid-sized, and they are myelinated, and they're actually your preganglionic neurons in the autonomic nervous system, so it's to do with your autonomic, so your guts. So you don't need to worry about those right now. A fibers are by far the most numerous in the body. They are large and myelinated. Okay, because there's so many, they have 4 categories according to speed. A alpha, A beta, A gamma, A delta. So the A alpha will be your motor neurons to your skeletal muscles, obviously need to be your fastest neurons, and your sensory neurons for proprioception, because your reflexes need to be really fast as well. A beta, second fastest, mechanoreceptors, so your touch, pressure, temperature, or touch, pressure, vibration. A gamma are your motor neurons into your muscle spindles, and A delta would be your fast pain. So that pain that you know exactly where it is, that paper cut you got, that hangnail you have, right? And also cold temperatures are actually carried on A delta. Okay, so A fibers, that's most of the nerves in our body, most of the axons in our body are large and myelinated. So they gave them 4 different categories because some are the largest, the A alpha, so they're the fastest, whereas the A delta, not near as fast as the A alpha, but still way faster than B and C fibers. Okay, so just a little reminder of those. Let's go to the synapse since we just took all that. What's the difference between an electrical synapse and a chemical synapse? Sure, the electrical signal and the chemical signal. So at an electrical synapse, the action potentials actually go through gap junctions. Remember gap junctions in the heart? It's a gap junction contains 100 or so tubular connexons, which act like tunnels to connect the cytosol of the 2 cells directly. As ions flow from one cell to the next through the connexons, the action potential spreads from cell to cell. Gap junctions are common in visceral smooth muscle, cardiac muscle, as we learned about, and the developing embryo. They also occur in your central nervous system. We don't spend a ton of time on those, but we did learn about them in the heart, right? Chemical synapses is where we spend most of our time now because it's the neurotransmitters that we're interested in. So although the presynaptic and postsynaptic neurons are in close proximity at their synapse, their plasma membranes don't touch. So as you can see, they don't touch there. There's a space between them. That's called the synaptic cleft. It's filled with interstitial fluid, and because nerve impulses cannot conduct across the cleft, an alternate indirect form of communication occurs. A neurotransmitter affects the postsynaptic neuron, muscle fiber, or gland as long as it remains bound to its receptors. So we can see the chemicals here. They're going to be released into the cleft. They're going to bind to the receptor. When we look at perception, I think it's important that we remember, especially for things like gait control and touch, and when we start to learn in advance about neurological conditions. Perception is our sensory experience of the world around us and involves both the recognition of environmental stimuli and actions in response to these stimuli. Through the perceptual process, we gain information about properties and elements of the environment that are critical to our survival. Perception not only creates our experience of the world around us, it allows us to act within our environment. Perception includes the five senses: touch, sight, taste, smell, and hearing. It also includes what is known as proprioception, a set of senses involving the ability to detect changes in body positions and movements. It also involves the cognitive processes required to process information, such as recognizing the face of a friend or detecting a familiar scent. So again, if we're going to be consciously aware of something, it has to make it all the way up to the top of our brain. You know, otherwise we don't have a lot of perception of it. Like, you know, our hypothalamus deals with our blood pressure, but it's the silent killer because we're not generally aware of it. Okay, so perception is like awareness. Okay, and it does help us with our survival. So when people have diabetes mellitus and can't feel their feet anymore, they're at risk for diabetic ulcers on their feet, which can lead to septicemia and all kinds of problems, cellulitis and things, because they can't feel it. They don't have a perception of what's happening with their feet. Okay, so this is part of our neurophysiology, is we need perception to occur to really be safe in our environment. So when we lose one of our senses, others can take over, but we are at a disadvantage. All right, the parts of a reflex arc. So that, again, this is a bit of review. We have the sensory receptor and the sensory neuron, which in the text they have as two things, but we know the sensory receptor is the dendrite of the neuron, that when it says sensory neuron, that's its axon. So the receptor is the dendrite portion, the sensory neuron is its axon. The integrating center, we took spinal reflexes, so that would be in the spinal cord. Okay, but we do have other reflexes where that might be up in the brain or the brainstem. The motor neuron, okay, and then the effector. So name this reflex. Stretch. Good job, team. Name this reflex. Yeah, it's a reflex. Which one? Tendon. Yeah. Okay, so we have the Golgi tendon organ here, so it's the tendon reflex. If we go back to those, in the stretch reflex, what happens to the agonist in the stretch reflex? Does it contract or relax? Contracts. A stretch reflex causes contraction of the agonist. Okay, so the tendon reflex, what does it do to the agonist? Contract or relax? Relax. Yes. Okay, and we use this a lot because we deal with a lot of hypertonicities and tight muscles, right? So we use the tendon reflex a lot in a lot of our treatments. As you're learning the different advanced extrinsic techniques, you're going to hear more about it. Okay, whereas some of your like IMFS, okay, and Topement, as we know, can bring about the stretch reflex, which brings tone back into the muscle, causes slight contraction. All right, nociceptors are found everywhere in the body, but the brain, right. That's why they can do brain surgery if they just freeze the scalp and the bone tissue. They can, and the meninges, they can get into the brain tissue. Endorphins work in the brain. True or false? Absolutely true. They kicked in for me today when I was doing that intense workout when I came home and I was just dead tired and I was all happy. Enkephalins affect the synapse by using the descending pathway to block the impulse from coming up the spinal cord to the brain. True or false? Absolutely true. So endorphins and enkephalins are sometimes lumped together, but they do work differently. Enkephalins are actually blocking the impulse of pain from coming up. Okay? Endorphins actually change the way you have an emotional attachment to your pain. So there's a difference. Endorphins, you'd be still aware that something's going on, but it's not going to bother you because like you're in such a good mood, you know? So you're aware it's there, but it's okay. You've taken away that emotional attachment to the pain with endorphins, whereas enkephalins actually stop the impulse from coming up. Okay, so that's just a different approach to dealing with the pain. Slow pain, which is deep, throbbing, achy, and diffuse pain, is carried on which type of axon or nerve fiber? Boom, you guys know that one, the C. Okay, sometimes called the polymodal fiber depending on where you're reading, so I just included that there. Identify the cellular anatomy of the neuron. Okay, so let's just start with the real basic anatomy. What's the incoming portion? We have the cell body, yeah. We have the axon. We have the dendrite. We have the axon hillock. Good job. I like that one. We have a synaptic cleft at the end, yes. We have the synaptic end bulb then, right? So here we go. Cell body, dendrite, axon. I heard axon hillock. We have that initial segment in the axon hillock where the trigger zone is, which is where that first channel is for sodium. We have myelin and neurofibril nodes or nodes of Ranvier. We have the neurolemma, which goes around um, that's the cell membrane of the neurolemmacyte or Schwann cell. Thanks, guys. Get brain freeze for a second. We have axon collaterals, we have axon terminals, we have the synaptic end bulb and the vesicles where we have the neurotransmitter. Okay, name the reflex. Causes contraction of a skeletal muscle, the effector in response to stretching of the muscle. What's the reflex? Stretch. In response to a painful stimulus, you immediately withdraw. Which reflex? No, you withdraw. You're pulling it away. No. Flexor. Yeah. So you're pulling away with the flexor or withdrawal reflex. A feedback mechanism to control muscle tension by causing muscle relaxation before muscle force becomes so great the tendons might be torn. The tendon reflex. Okay, and synchronizes the extension of the contralateral limb with the withdrawal or flexion of the stimulated limb. No, that was the painful stimulus. This is the other one, contralateral, right? So you step on the tack on the left side, you need a contralateral to stimulate the right side to catch you so you don't fall down. Okay? So we have the stretch, the flexor, the tendon, and the crossed extensor. All right. So what does occurring or located on the same side? What word? Good job, ipsilateral. Involves more than 2 types of neurons and more than 1 central nervous system synapse. We're referring to reflexes. Polysynaptic. Good job. And a reflex pathway having only one synapse? Monosynaptic. Okay, so these were terms for reflexes, right? So if it's on the same side, it's ipsilateral. If it's on the opposite side, it's contralateral. If it has more than one synapse, it's called polysynaptic. There's only one that's monosynaptic, and we know that's the stretch reflex, right? Considered monosynaptic. All right, can you recognize these definitions? The proprioceptors in skeletal muscles that monitor changes in the length of skeletal muscles and participate in stretch reflexes. Spindles. Located at the junction of the tendon and a muscle, these proprioceptors protect tendons and their associated muscles from damage due to excessive tension. Boom, GTO. That's the easiest one you've been able to type in so far, GTO. Okay, good job. Okay, more definitions. A sensory nerve ending, primarily located within the muscles and tendons, which receives stimuli pertaining to movements and position of the body. No, it's the general name. Proprioceptor, good job. I saw it pop up there. Yep, proprioceptors. The restriction or arrest of a function or specific activity. I hate this definition, I will just tell you now. Right? Yeah. It's inhibition. Okay? That's what it's not. Yeah, I know. It's a dumb definition. I got to change that. So inhibition. This next one, don't even read it. This is referring to, let's see if you can get it. This is referring to the outside of the cell membrane. Okay? An increase in the positive charges normally present at the surface of a nerve cell membrane. Right? Because this is the opposite of what you would think. Yeah. It's hyperpolarization. Yeah, because it's referring to the outside, right? We're always, whenever we're talking about depolarization or hyperpolarization, the textbooks refer to the inside. And I found this definition because it's referring to the surface, so the outside. And I thought, well, that'll make them think. But personally, I don't like it either. Okay, so, and then the elimination or neutralization of polarity is depolarization. Okay, so just some different definitions. Don't worry, they're not on the test. But it's just there to make you think. You know, sometimes you see a different definition and it makes you think. And I think sometimes it can be a learning moment if you let it. But yes, on the test, it's plain language. Trust me, okay? But here I just wanted to make you guys think. But the answers are right here, okay? And this is going up right after class. A peripheral nerve organ that receives and transmits painful sensations. Nociceptor. Good job. A receptor that responds to the stimulation of mechanical pressure. Mechanoreceptors. That was plain language. A skin area supplied by sensory fibers of a single spinal nerve. Oh, y'all got that one. Dermatome. Good. A medication that relieves pain without affecting consciousness. Analgesic. Good. Okay. When it affects your consciousness, they're called opioids. Oh, booze. Sure. Have a glass of wine. Okay. Good one. Okay. We already went over the answers, so forget about this part. But how do you feel about your level of understanding? If it's good, smile. If it's bad, what are you going to do about it? And I thought I would include this learning pyramid. Okay, so at the lecture, about 5% of understanding. Once you read it, another 10%. Audio visual, so pictures, watching videos, 20%. Once you can demonstrate it, so you get to demonstrate, okay, 30%. Once you discuss it with somebody else. We're talking about not memorization and short retention time. We're talking about true understanding. Okay, if you can discuss it, you get another 50%. If you can practice doing it, okay, 75%, and then teaching others, 90%. So to really understand something and the way the brain works, you can't just rely on one of these. You have to do a lot of them. It's why studying in groups is good, because you get a lot of these top ones. You know, a lot of people say, oh, I like to study on my own, but then you're still in these lower ones. It's when you work with others, you do a lot better, okay? Well, that's awesome, Jessica. Like, that's awesome, right? Moving on up. So learning is the act or process or experience of gaining knowledge or skill. Understanding is comprehension, okay? So there's, you know, something to think about there. Okay, so understood and forgot. I like that one. That means you didn't get it into the long-term memory, so you got to keep playing with it. And this is why we do repeat this stuff in advanced, right? Okay, and you get it repeated in practical, and then you're going to get it in advanced practical and physiology and pathology. They're all reviewed again because we want them to be in long-term memory, right? So you can always apply your techniques with knowledge, which is important. Okay, because you don't want to apply them incorrectly. All right, we're going to take a 5-minute micro break, and then I just have a short PowerPoint to remind you about your techniques, okay? And a little bit of other stuff. Okay, and then both of these PowerPoints are going to go up on Moodle for you after class. So 5-minute break. Yeah, Maria, which lecture are you asking about? Like last seminar weekend in physiology, we covered muscle tissue, mostly the cell, and muscle structure. And in pathology, and then we covered the muscular pathologies or soft tissue pathologies, and we finished them up at the evening class a couple weeks ago. Or was that last week? Whatever it was. All right, well, I'm just going to go on now. So in this one, it's just a little bit of review to make you think about your techniques a little bit, okay? All right, at synapses, only one-way information transfer can occur from a presynaptic neuron to a postsynaptic neuron or to an effector such as a muscle fiber or a gland. Okay, so we see the neurotransmitter will be released and then affect the postsynaptic membrane, be it nerve, muscle, or gland. The synapse formed between the axon terminals of a motor neuron and the motor end plate of a muscle fiber is known as the neuromuscular junction, which we also just took about in physiology, right? So we have the A alpha motor neuron coming to the motor end plate of the muscle cell membrane, the sarcolemma, and there are receptors for acetylcholine. When a postsynaptic neuron depolarizes, the effect is excitatory. If threshold is reached, one or more nerve impulses are initiated. By contrast, hyperpolarization has an inhibitory effect on the postsynaptic neuron. As the membrane potential moves further away from threshold, nerve impulses are less likely to arise. So they're called an EPSP, excitatory postsynaptic potential, or an IPSP, an inhibitory postsynaptic potential. Okay? The neuromuscular junction is always excitatory, so it's always an EPSP. The only neurotransmitter released at the neuromuscular junction is acetylcholine. Okay, so when you want to inhibit a muscle, you have to inhibit the A-alpha motor neuron all the way up in the spinal cord. And then there are fewer impulses coming down to the neuromuscular junction, so less muscle contraction. Okay, so when you have the agonist contract and the antagonist is inhibited, okay, the muscle itself here at the neuromuscular junction, just less is happening because up in the spinal cord, this motor neuron is being less stimulated. It's being inhibited. Okay, so neuromuscular junction is always excitatory. The only thing you can do is turn it down. Okay, you can't, inhibit the actual neuromuscular junction because the only neurotransmitter released here is acetylcholine and it's always stimulatory. Okay, so we have about 125 substances that are either known or suspected neurotransmitters. So in other words, they don't know a lot yet. Most neurotransmitters are synthesized and loaded into synaptic vesicles in the synaptic end bulbs close to their site of release. Okay, so at the neuromuscular junction, we know acetylcholine is excitatory, and that's the one we talk about the most. And then the inhibitory neurotransmitters we talk about the most are glycine and GABA. Okay, glycine tends to be more used in the spinal cord and GABA more up in the brain. Okay, so again, we see these in inhibitory neurotransmitters are in the central nervous system. Okay, and yeah. So stimulatory techniques then in massage cause excitatory postsynaptic potentials or stimulation of the motor neuron to the muscle being stimulated. So things we use for that would be like tapotement, IMFS, and doing things very quickly like fast stripping, etc. Okay, so when you want to cause stimulation, you either do it really fast, okay, or you use things like tapotement and IMFS, which are both done quickly, okay, because you want to cause a fast stretch on the muscle cells, which would cause the stretch reflex, causing the muscle to contract. Okay, so anything that you're doing that you want to be stimulatory. Now, we don't come across a lot of weaknesses in massage or in people other than those that are maybe from a neurological condition, which all the stimulatory techniques in the world aren't going to help. But when we do come across them is when a nerve has been impinged further up or more proximal. Okay, so for instance, if your brachial plexus gets impinged, you get a weakness in your hand. You don't have as good of a grip, right? You get some numbness, some tingling, etc. So you can get the brachial plexus where the hypertonicity is, say in the pec minor, and release that. But then you could do some fast stripping and stuff of the muscles that serve the wrist to get that strength back up. So sometimes we will do stimulatory more distally for muscles that haven't been receiving as much stimulation because of a nerve impingement more proximally. Okay, so remember, stimulatory techniques that we do are going to cause an excitatory postsynaptic potential, stimulation of the motor neuron, which then obviously stimulates the muscle. Most of those techniques are done quickly to elicit the stretch reflex and induce tone. Inhibitory techniques, then, we want to cause an inhibitory postsynaptic potential. So we want to cause inhibition of the motor neuron to the muscle being treated. So, for example, your isometrics, which you'll learn later, techniques are done, and other techniques which are done slowly with a stretch on the tissue. Remember that stretch is the most important thing. So things like slow stripping, okay? So to get an inhibitory technique to work, it's actually done in two ways. You elicit the tendon reflex, which we know causes inhibition of the motor neuron to the agonist muscle, okay? But we also get muscle spindle adaptation. So what happens there is when you are doing techniques that are very slow and a nice slow stretch on the muscle, it bunches up a little bit and takes some of that pressure off of the muscle spindle, causing the spindle to fire less, okay, because remember it's monitoring that stretch on it, so it fires less, and you do that enough times and it adapts to that. And so you have less tone into a hypertonic muscle and it gets back to normal tone. Okay, or you have the tendon reflex fire enough, causing inhibition to the motor neuron that serves the muscle, and that causes the muscle spindle to have less pull on it, and again it will adapt to the new resting length. Okay, so whenever you're trying to do things that are inhibitory, you slow it down. You want to take that pull off of the muscle spindle, and we use the tendon reflex to do that because that's going to cause that inhibition. That takes the pull off. But any of the techniques we do, we want to do deep and slow so that we can get that bunching up in front of our hands as we're working with a slight pull on the tendon. Okay, so all of your IPSPs will get the muscle to adapt. So the tendon reflex is one that we use, and isometrics have a direct effect on the GTO, causing the reflex inhibition. So we know that directly we cause it there. With the advanced extrinsic techniques, if you have a stretch on the affected tissue, okay, you'll have an effect on the GTO. So things like slow stripping, strain counter strain, and again you'll get that muscle spindle adaptation because of the GTO firing. Okay, so you want to be able to get that that stretch onto it, which feels like more of a threat, and then you do these deep techniques into there. Okay, so what is muscle spindle adaptation? Big words, right? It's a temporary reduction of tensile strength of the muscle's connective tissue, may cause the muscle spindles of the proprioceptive nervous system to reset to a less sensitive level. This in turn may allow the resting tone of the muscle to reduce. The main function of muscle spindle is to measure muscle length, how much a muscle is being stretched. Either sudden or prolonged stretching of the central areas of the intrafusal muscle fibers stimulates the nerve endings. The resulting nerve impulses propagate into the central nervous system. Information from muscle spindles arrives quickly at the somatic sensory areas of the cerebral cortex, which allows conscious perception of limb positions and movements. At the same time, impulses from muscle spindles pass to the cerebellum, where the input is used to coordinate muscle contractions. Okay, so the muscle spindle's involved in a lot, and that's why it can get messed up depending on how you're using your body. And so when we get it to adapt, we want it to adapt to a new resting length. Okay, so to elongate. That's our intent with that. So don't forget, we have two types of effects with all of these things that you're learning, mechanical and neurophysiological. The mechanical effects include changes in the structural integrity of the tissue, local thermal changes, passive vascular changes. Extrinsic techniques with mechanical effects of altering the structural integrity of the muscle tissue are quite effective in treating contracture. Okay, so if you're working with somebody with a neurological condition, okay, so their nerves don't fire correctly, say somebody with advanced MS, trying to do anything with a neurophysiological intent, probably not the best because their nervous system doesn't react properly anyway. So you would stick to mechanical effects. So your intent of treatment, remember you've heard a lot about intent, would be more mechanical then. You learn about neurophysiological effects, and there's two primary neurophysiological effects: increased resting tone and decreased resting tone. Thus, the stroke types are referred to as stimulatory and inhibitory. Okay, so very effective in treating hypotonicity and hypertonicity. Okay, so that was just my little boost to you to help with thinking about the neurophys that we've learned and applying it to what you're learning now in practical and applying those advanced extrinsic techniques. Did you guys find that helpful? Sort of summed up in one little PowerPoint? Yes? Okay, so I'll be loading this right after class as well, and you can look it over. What it's trying to do is, yeah, put the two together. You know, you took neurophysiology and now you're going to have it in practical, right? And I tried to take everything and put it into one PowerPoint so it's not very specific for each of your techniques, but you're learning that right now. But I just wanted to tie it with the neurophys we've taken. Okay, so definitely play with that. I'll load it right after class here and you'll have it to use to go over. Okay, so definitely use it as a review. So just a reminder, if you go on Moodle, you'll have your study guide. Okay, you're welcome guys. You'll have your study guide for Neurophys. We still have one more class for Neurophys before the exam, and that will be June 13th. Okay, for the June 13th class, I want you guys, I'll have a few PowerPoints here to refer to. I need your questions. So we'll use the study guide at that point and then any questions you have from that. So it might not be a very long class if you guys don't come with questions. If you're comfortable, then I'll be comfortable and that'll be that. But we do have one more class June 13th, but I'm going to leave it up to you guys to bring your questions. So do some studying, figure out what you want to review, and let me know. If you can let me know ahead of time, even better. I can have some stuff ready. Okay, that's all I have for tonight. It's on everything. So if you look at the study guide, it has everything right from the spinal cord, spinal nerves, meninges, got a little bit of everything on there. Okay. Okay. All right, I'm going to stop